Hello, everybody. Thanks for coming. So many people here makes me a little bit nervous. But hello, I'm Luna, and this is Naldo, Tina, and Angel. Together, we are going to present our presentation. Tina and I, we are sophomore students, and Naldo and Angel are junior students. Our project, Lucaparon, it's an auto-regulated system with both feed-forward and feedback controls. We are from the National Yang-Ming University from Taipei, Taiwan. First, I will give you our outlines for this presentation. I will first introduce our team members and our motivation. Then comes our current approach and current problem. And then is our approach with design, engineering, and results. The ending will end with the achievement and our conclusions. So here comes our team members. And we have two faculty advisors from our school's Center for System and Synthetic Biology, some graduate consultants. And we are mainly undergraduate students with backgrounds in medicine, dentistry, life science, computer science, engineering, and bioinformatics. So what is Lucaparon actually doing? Lucaparon, a glucose-based synthetic operation system. You can also say that it is a glucose-induced insulin secretion system with regulated controls. So about diabetes, I will first give you some data from the World Health Organization published in 2006 that every year around 3 million people died from diabetes worldwide. And around 180 million people are having diabetes. And it is predicted that in 2030, the number of diabetes patients number will double to 360 million people. So the glucose concentration in our blood sugar is definitely a very important issue nowadays. And about diabetes, there are actually two counteracting forces in our body. One is the glucose concentration, and the other is the insulin concentration. Once the glucose concentration raises here, our body will automatically secrete insulin that lowers the glucose concentration in our body to maintain health. So what's the current treatment for severe diabetes patients nowadays? We are usually providing their insulin. Insulin can definitely lower glucose concentration from a hypoglycemia state to a normal range, but once it's not regulated properly, it will also fall down to this hypoglycemia state, which may cause coma or even death. So to maintain homeostasis, we have to consider a third component, the insulin-degrading enzyme IDE. IDE is actually existing in our body. And there's a time lag between the insulin peak and the IDE peak. This time is sufficient enough for insulin to lower glucose concentration down to this proper range here, before it is degraded by IDE. But with a diabetes patient who is treated with extra insulin, this regulation system is not that normal. So we are wondering if we can get a smarter cell which can give us a perfect regulation. Yes, definitely. From the aspect from synthetic biology, we have a design for the cell. That's Naldo. Let's welcome Anne.
Okay, and thanks Luna and Naldo. Now let me tell you how did we design an auto-regulated system to prevent hyperglycemia and hypoglycemia. Well, in order to prevent hyperglycemia, when high glucose concentration signal input into our system will be activated and generate insulin to decrease the glucose concentration back to normal level. But if the secreted insulin continue to express, the glucose concentration will be too low, and which leads to hypoglycemia and then coma, which is dangerous to patient life. So, so we need an insulin-degrading enzyme IDE to degrade the secreted insulin and prevent it from further decreasing the glucose concentration and also prevent hypoglycemia. But if the IDE and insulin express at the same time, insulin will not take effect and may be degraded by insulin-degrading enzyme. So there must be a time delay between the expression of these two molecules. And in order to introduce a time delay into our system, we use a feedforward-like circuit into our design, and the circuit can create the time delay. Well, let me introduce the circuit. When the S starts to express, When S start to express, Y molecule is stimulated and also start to express, but Z molecule is inhibited at this time. Then, as the S molecule start to decrease, Z molecule is not inhibited anymore and also start to express. But when the Z molecule start to express, Y molecule loses stimulation Y and start to decrease. And for a period of time, Z molecule also loses stimulation Y and start to decrease. Sorry. And then you can see that there's a time delay between the pulse Y and the pulse Z. And if we change Y to insulin and Z to ID insulin degrading enzyme, we can successfully create a time delay between the expression of the two molecules. And in our experiment, we change S molecule to glucose, Y molecule to CNRHS cell, which express together with insulin, and the Z molecule to ID insulin degrading enzyme. When high concentration of glucose signal input into our system, Y molecule together with insulin are stimulated and start to express. But because we design inhibition binding site here, high concentration of glucose will lead to some molecule bind to this inhibition binding site, and Z molecule insulin degrading enzyme will be inhibited at this time. Then when the glucose fall below the threshold and the inhibition site is not occupied, and the Z molecule insulin degrading enzyme start to express and to degrade insulin, prevent it from further decreasing the glucose concentration. And now let me take an overview of our circuit. When the glucose concentration starts to increase and reach the threshold, insulin and the CNRHSL is stimulated and start to express. Then, when insulin successfully decreasing the glucose concentration back to normal level, the insulin degrading enzyme is not inhibited anymore and start to degrade insulin and prevent from further decreasing the glucose concentration. Then ID will be degraded by its protein auto degradation, and our system finish their work, help bring the glucose back to normal level. And later, Tina will tell you more detail about our system.
Thanks, Nado. I'm Tina. How do we know our system works? And to know our system works, we must embed glucagon systems in expression cells and have a reporter cell. We chose mammalian muscle cells as our reporter cell, for it is the main insulin target cell in human body. We chose E. coli as our expression cell, for it is easy to operate, and we just want to prove of concepts. Glucagon systems are composed of two devices. The two devices are composed of several parts. I will tell the details of the parts later. Now let's focus on how the two cells interact with each other. When expression cell sees high concentration of glucose, it will then generate insulin. Insulin then binds to insulin receptors on the reporter cells and helps the intake of glucose. Now, let's see the molecular model of insulin generation device. High concentration of glucose is transported into the expression cells and leads to the phosphorylation of unbound molecule. Phosphorylated unbound then bind to per C and lead to the transcription of insulin. SP here means twin of the cell. After insulin is translated, insulin will alpha chain and beta chain will form disulfide bonds and be transported out of the cell because of SP. Besides insulin alpha chain and beta chain, Insulin generation device also express signal HSL. Signal HSL connects insulin generation device to insulin degradation device. Because insulin and signal HSL expressed at the same time, we can view HSL. This Barbie model shows the process we combined our insulin generation device. The yellow background highlights our contributions to MLT power registry. Now let's turn to insulin degradation device. Since insulin degradation device is much more complex than insulin generation device, we must add a NOT and B gate in front of the insulin degradation device. In other words, insulin degradation device is activated only when glucose is in low concentration and insulin exists. Let's see how insulin degradation device works in high concentration of glucose. High concentration of glucose will lead to the phosphorylated of amide molecule. Amide molecule then binds to amide binding site and degradation device and thus blocks transcription. Let's see how insulin degradation device is work in low glucose concentration. When synthesized insulin lowers the concentration of glucose, the amide binding site will not be blocked anymore, and the signal HSL protein complex binds to promoter signal HSL and leads to the transcription of IDE. Again, signal peptide here helps IDE be transported out of the cell. IDE then degrades insulin and brings insulin and glucose concentration back to normal level. Again, this Barbie model shows the process we make out our insulin degradation device. The yellow background highlights our contributions to MLT power registry. Besides that, we use gate-free assembling to make out our parts. And next, Andrew will tell you our results.
Thanks, Tina. I'm Angel. To test our system, first, we want to make sure that our expression system can work with our reporter system. And this two system has been mentioned before. So, we put our mammalian reporter system into the E. coli-free medium, EFM, which is a medium that has been cultured with E. coli. Then, the E. coli has been removed, and the E. coli secretion remained. And we found that the growth of mammalian reporter cells in the EFM are approximately the same as the control. After we make sure that our mammalian reporter system can work with our E. coli expression system, and then we want to know whether our mammalian reporter system can be activated by functional insulin. From the experiment result, you can see that glucose concentration dropped 5% in 20 minutes, which means that our mammalian reporter system can uptake glucose after induced by insulin. And then, depends on our system, the insulin generation device can be induced by glucose. So, we add an EYFP behind our promoter, POMC. If the POMC is induced, it will activate EYFP. And we can see the fluorescent light under the fluorescent microscope. In this table, you can see that if there's no glucose, there's no fluorescent light. If there's 1% glucose, you can see the fluorescent light under the microscope. After, we make sure that our system can be induced by glucose. And then, we want to know how long did it take to induce our system. From the experiment data, we draw a time versus YFP intensity graph. You can see that glucose induction increased 10 times after 6 hours. And then we want to know whether our insulin generation device can express insulin or not. With the Western blot result, you can see there's a band around 10 kilodalton, which is the size of insulin with signal peptide. Thus, our insulin generation can express insulin. Next, Luna will tell you about our conclusion.
Okay, here am I again. Firstly, I will say this. This is a proof of concept system that can auto-regulate the glucose concentration. We have 2 feedback controls, the black lines here, and 1 feed-forward loop. And we are the first team to implement the type 3 incoherent feed-forward loop in our system. And we also have constructed a new gate. We have combined the NUT and AND gate together. And in contrast to the current treatment, in which you have to replenish insulin all the times, the best thing of our glucagon is that it is a self-propagating system. You don't need to give them insulin all the times, and it will maintain the glucose concentration in a normal range. Our result has been accepted in the 2007 International Conference on Bionano-Science, and we hope that in the future, our system can be implemented into the mammalian cells and improve the quality of life of the diabetes patients. Thanks for all the fundings from our school and the Ministry of Education in Taiwan, and a special thanks for these people here, and of course for your attention today. Your suggestions and comments are more than welcome. Thank you. Here are other team members, and if you have any questions, please raise your hand.
University iGEM team from Taipei, Taiwan, you can see here. And I'm Clio. I'm going to introduce the Back to Kidney, our project, and I'll give you the overview of it. Well, Back to Kidney is a bacterial-based microdialysis machine which can improve the quality of life of chronic renal failure patients. And you can see our team members, including faculty and graduate consultants, and of course, undergraduate students is here, okay? And our beautiful campus, you can see here as photos. Okay, first of all, I'll talk about what's chronic renal failure. It's a syndrome that may be caused by many diseases. That means kidneys no longer remove metabolic waste and maintain the balance of body fluid efficiently and well. So, and these patients rely on hemodialysis or kidney transplantation to maintain their lives. Well, hemodialysis here's many drawbacks of it. Well, it's costing and inconvenient for the patients. They have to stay right beside the machine for hours every 2 to 3 days. And they have constriction on diets. They suffer injuries on blood vessels. Well, it ultimately causing uncoverable damage. So, is there anything we can do for these people? They live such inconvenient lives. Someone did invent a so-called portable hemodialysis machine. As you can see here, the real size of it is about this big. So it's not really portable enough for someone who want to come to iGEM Jamboree from Taiwan, so far away. Okay. Here you can see. This is our solution for making we're considering the possibilities of making the microdialysis machine tinier and easier for the hemodialysis to use. And here's 4 characteristics we want it to have: portable, time-saving, self-regulation, and of course, abilities to remove waste. And here you can see the comparisons. We compare our Back to Kidney and traditional hemodialysis. Here's the 4 main components of the Back to Kidney. And first I'll tell you is pH sensor serves as a switch. it will turn on the whole circuit we design. If it sends the environmental pH value is around 7 or 8, indicating that our bacteria is arriving in the small intestine. And the second, attachment. Attachment, well, this group will enable the bacteria to stay right on the small intestine epithelial cell, providing sufficient time for it to work. And what's its work? The work is to remove the waste we don't want. And you can see here the 3 waste we want it to remove, respectively. They are urea, guanidine, and phosphate. And why do we choose these 3? Because they will have more toxicity if accumulated. For example, abnormal phosphate concentration in our body will cause pruritus. It's a disease makes you feel itch. And finally, our splendid time regulation. It will tell the bacteria when to leave and detach from the small intestine, leave our body. Well, here's our 4 components and overview, but before you go on the details, I'd like to tell you, you can see the 4 small icons on the upper right corner, and they indicate pH sensor, attachment, waste removal, and time regulation respectively. So if you get lost in the slides, just seek for their help, don't worry, and enjoy our presentation.
Thanks, Lyle. Hello everyone, I'm Laura. I'm going to talk about initiate a whole project. Once we determine to do something, it's done at right time and right place, and all if done at proper spatial and temporal situation, a thing of good intent may bring good effect. So here's the problem. Where does the bacterium work the best? While removing the metabolic waste from the renal failure patients. Well, the location is not in the lymph fluid or blood flow, since that might lead to candidemia or many other systemic diseases. After evaluating all the candidates, decided to let our microdialysis machine, back to kidney, work only in the small intestine, where there is an environment of high pH around 7 to 8. That is to say, we prospect on our pH sensor since the high pH condition and express the dialysis gene expression as our goal. In order to find what kind of genetic component with desirable pH sensor, review the left literature about the correlation between pH condition and the bacterium living. We found a system regulating the ion exchange between the external environment and the E. coli host cell. A system and its proven function are not shown on the slide. There are some channels transporting proteins outside, some others transporting sodium ions inside. Or there are still some special ones, named the NHAA, called transporting the mentioned ion on the opposite direction. This channel delicately cooperate with each other, keeping the cytosomal concentration of sodium and protein at the balanced level, which allows the bacterium to live well. If the external concentration of protein is much lower than the one in the cytosol of E. coli, it means this E. coli encounter a high pH stress. There will be a tendency in those channels transporting proteins outside and sodium ions inside. However, to prevent the cytosomal sodium ion level from getting too high, there's another mechanism involved. Besides many other physiological characteristics, a sodium ion is a component of the NHAR protein sodium ion complex, which plays a role as a driving promoter of NHAA. On this amazing promoter, the gene NHAA will be expressed where there are abundant amount of sodium ion in the cytosol of E. coli. In our project, we apply this promoter to be the manpower switch, making sure our microdialysis machine, back to kidney, work only when arriving in the correct place, the small intestine. Most of the regulating circuits mentioned later are put at the downstream of this promoter, PNHAA. Now to Jordan tell us how our microdialysis machines stay and live our small intestine.
Okay, thanks Laura, I'm Jordan. So, to remove the waste in the small intestine, first we need a pH sensor to let our micro machine to work at the right place. Also, we need another mechanism to let our micro machine to attach to the small intestine. That is what I'm going to tell you, attachment. So to attach to the small intestine, we must express a protein which can mediate binding between our micromachine as well as the epithelial cell of the small intestine. And as you can see here, it is our binding circuit of a fusion protein, which can be separated in the three parts. The first part is LPP, lipoprotein signal peptide, is the signal peptide which can target our fusion protein to the outer membrane of the E. coli. And the second part is OMP-A, outer membrane protein A. We use its transmembrane part to cross our fusion protein to the outer membrane of E. coli. And the third part is FIN-H, fiber protein H, is the outer membrane part of our fusion protein, and it can mediate attachment between cells and bacteria. And the whole circuit is under the regulate of pH sensor, pNHAA. So when the micromachine sends a suitable value of pH in the intestine lumen, it will give express of the whole fusion protein, and our micromachine can attach to the small intestine. However, why we choose FIN-H as our attachment material? Well, that is because FIN-H can mediate mannose binding between bacteria as well as the epithelial cell. And because mannose is very abundant on the epithelium of small intestine, so overexpression of FIN-H can let our micromachine attach to the small intestine. So, our micromachine has an ability to attach to a small intestine. However, when its work has been done, it has to be removed from the small intestine. So we design a protease cleavage site inside the fusion protein, and also a circuit encodes a protease and under-regulated by a time-regulating promoter. So when the time is up, the protease will be released and cut the fusion protein. So this machine will detach from epithelial cell and then be removed from the small intestine. So far we have a pH-mediate attaching mechanism as well as a time-regulated detaching mechanism. However, you may ask, how this tiny machine know when to detach from the small intestine? Well, that is through time regulation, and our team member will tell you how we regulate the time.
Hello, I'm Edward, and I will explain the time regulation design of our project for you. There is a time between attachment and detachment, and since we want to be able to specify this time period, we create a timer. The timer consists of three parts. The starter, which sends pH value as an input signal. The timing part, which uses an oscillatory system to keep track of time. And the stopper, which can produce some output after a specified amount of time. At first, we want the bacteria to start the timer when the bacteria attach to the small intestine. And since the bacteria could use pH sensors and HAA promoter when it attach, we also use the same promoter as an input to start our timer. At last, as for the stopper, we stop the timer by setting a threshold. As the protein produced by the oscillatory system accumulate and reach a certain threshold, the activity of the output promoter will suddenly change and produce the protease as an output. In our timer, we have created a so-called oscillatory system in which we could connect different oscillators for different purposes. For example, we could use two different oscillators we have created in our project. One is the cyano oscillator, which has a longer time period. And the other is the relaxolator, which has a shorter time period. The oscillatory system was created so that any type of oscillator could be implemented as long as it conformed to the system. The system only requires that there exists a promoter which has an oscillatory expression pattern. The first oscillator we create is a three-component oscillator taken from cyanobacteria. It naturally occurs in cyanobacteria and could have a period of 14 to 60 hours. The second oscillator we create is a two-component relaxation oscillator, which we coined the relaxator. In addition to just making an oscillator, we also make a tuner to tune the period of the oscillator and a synchronizer to synchronize the oscillator between different cells. After specifying the time between attachment and detachment, we can then let the bacteria start to remove the 3 unwanted waste, and Wendy will detail this part for you.
Hi, I'm Wendy. I would like to introduce the urea and guanidine removal part to you. The researchers have found the biosynthesis of guanidine is resulted from the transaminations of arginine from urea. So the rising level of the urea may cause the guanidine accumulation in renal disease patients. With both concentrations rising, the poor renal disease patient will fall into coma and massive hemorrhage. So to remove guanidine and urea do become our prime target. And by reviewing related information, we find that both urea and guanidine can be absorbed by bacteria. And this method could have a higher efficiency and higher specificity comparing to the method of hydrolysis and absorption. And this is a diagram to show how can our microdialysis machine works in a small intestine. First, the waste present in the intestinal lumens will be taken by our machines through the active transporter, represented as the yellow channels one. And this will cause the osmolarities in the intestinal lumen to decrease. So the urea and guanidines in the intestinal cells will diffuse into the intestinal lumen and taken by our machines again. As a result, the urea and guanidine present in the renal disease patient blood will become lower and lower. In order to accomplish our goal, we designed the circuit as below. Initially, the urea will slightly diffuse into our machine, as shown in this figure. And first, we'll build a promoter called promoter NHAA, which is a pH sensor mentioned before. And it will express the downstream urea, which is a urea binding protein and form a complex with urea. And this complex will bind to another circuit, to the promoter urea-D, and express the downstream urea transporter and guanidine transporter to express more on our machine surface. Therefore, the urea can be taken more, and the guanidine can be taken more. And what I want to mention specifically here is that the urea taken by our active urea transporters can again bind to the urea and urea, which can enhance the whole circuit and is a positive feedback. And in a closer look to our transporter gene, the urea transporters expressed in the helicobacter, which is a function to fluxing urea. And the guanidine transporters is a homolog from human cation transporter 3 from K12 E. coli, which has a functions to fluxing guanidine. Okay, now let us welcome Ariel to another important part, the phosphate removal.
Hello, I'm Ariel. The next waste we are going to remove is the phosphate. However, what is the regulation device we need actually? First of all, we hope it can absorb the phosphate when the external pH is high and the external phosphate level is high. What's more, we hope it to stop absorbing the phosphate when the external pH is low or where the external phosphate level is low, because the phosphate is also an important source for the ATP and DNA. So to achieve our goal, except the pH sensor we have before, we still need two more abilities. They are the ability to absorb external phosphate and the ability to sense the low external phosphate level. So now we are going to choose the proper element from E. coli. In E. coli, there are two systems responsible for the absorption of the phosphate. They are the PST system and the PIT system. And because of the higher affinity of the PST system and the lower velocity, we decided to choose it to build our construct. And in E. coli, there are another regulation pathway to transfer the phosphate into the polyphosphate, poly-P, to store it so the E. coli can keep absorbing the phosphate. So we also choose the enzyme PPK, polyphosphate kinase, to build our construct. It's responsible for the transformation between the ATP and polyphosphate. So as I mentioned before, we designed our circuit with the pH sensor and PPK for phosphate storage and also the PST for the phosphate transport. So another ability is to sense the external low phosphate level. And when the E. coli is under the low concentration stress, the protein 4R will going to phosphorylate the phobe, and the phobe will going to regulate the phoregulon. And the PST system will express up to the membrane to absorb the phosphate. So we can say that the phobe can sense the low concentration of the phosphate. That's the reason why we decided to choose the promoter of it to build our construct. However, the function of phobe seems totally opposite to our purpose, which is expected to sense the low concentration and then decrease the absorption. So we must do some modification. So here's our circuit design. When the pH sensor is turned on, the PPK and the PST will be expressed and keep absorbing the phosphate. And when the phosphate concentration becomes lower and lower and attain a threshold, the promoter phobe will be turned on, and then the theta will be expressed and going to inhibit the p-tate. So the expression of the PPK and the PST will be inhibited too. So our tiny machine can regulate the phosphate concentration in the intestine. So now for Ruby.
There's 2 minutes for questions.
Thank you. Hi, I'm Ruby, and now here's the experimental and model results. First, in the pH sensing part, the testing result shows that our high pH sensing promoter, the PNHAA, can indeed activate it when the pH level is high. So it can be used in our circuit to help to target to the small intestine lumen. And in the attachment part, the adherence assay shows that the Finmage protein indeed enhances the adherence ability of the bacteria. Therefore, the E. coli equipped with the Finmage protein can be better adhere on the cell after several times of PBS wash, and therefore release the GFP signals. And in the urea removal part, the urea sensing protein, the URER, can indeed bind in with the urea and therefore activate the downstream promoter of URED in our model. And also, the urea transporter and the guanine transporter can also uptake the excessive waste in the environment in our model. And in the phosphate removal part, we first measure the phosphate removal efficiency of the wild-type E. coli. And from the experiment result, we can see that the E. coli can uptake the phosphate itself, but it cannot regulate the amount, especially when the phosphate level is too low. So we put a low phosphate sensing promoter, the promoter of phobe, into our circuit. And in our experiment result, it shows that it indeed can be activated when the phosphate level is too low. And for the time regulating the reoxalator, the model shows that the 2-component oscillator can have oscillation through time. And we've done the reoxalator assay, but we are still working to make it have more stable oscillation. So far we've built the 69 BioBricks and 5 devices onto the BioBrick registry, and we've successfully verified some of the BioBricks. So in conclusion, our back-to-kidney can work at the right time, right place, remove the waste we don't want, and detach in time. And what we are going to do in the future is to finish verify all of the functional parts in vitro, and then put all the parts into one appropriate bacteria, and finally put this bacteria into animal model to test whether it works or not. And finally, thanks for these people providing us experiment materials and useful suggestions, and the National Yang-Ming University and the Ministry of Education for funding. And thanks for listening. If you have further interest in our project, please visit our official wiki site or come to a team member of us. Thank you.
Okay, hello, ladies and gentlemen. I'm Nora. We are going to tell you a legendary story of our project. Speedy Back! And this is the outline of our presentation. First, who are we? Once upon the year of 2010, we, a group of iGEMers, come from the Earth, Asia, Taiwan, Taipei, and National Yang-Ming University. This is our beautiful campus. Second, what are we trying to do? Our goal is to provide a faster assay system for exploring the design rules for synthetic biology. And the third, what do we want to do that? The current truth is that all the iGEMers around the world are trying hard to solve these problems, but however, they realize a terrible truth. That is, in order to work on synthetic biology, we need both biological parts and also design rules. Moreover, we already have a registry of biological parts, but where's the registry of design rules? And also, therefore, most iGEMers around the world are still doing that black box design, but because there are no registry of design rules, how to design a black box? Is the black box black enough? Again, where is the registry of our design rules? So we all need that the major need of synthetic biology is all about the design rules. So we can design many parts of biobricks we want just by using software, and also can combine them into anything. Just like the IC circuits, we can combine the individual parts into a complete functional circuit, but the combination needs rules. And also, every iGEM teams of every year's competition will register all their parts on these website NetPart registry. And if we want to combine these individual parts into a biological system, it also needs rules. So the lack of rules causes limitations as the development in synthetic biology. So for combining the biobricks needs more knowledge about the biological system. And for understanding the rules in the biological system, it needs time. So it will take you many years to find out the design rules for synthetic biology. It's not what we make. It's not only how we make it, it's how fast we can make it. So how you make is our feedback. Thank you. Conventionally, most efforts on synthetic biology work focus on the transcriptional control. That is to say, most of us pay attention to the protein expression once the DNA starts to work, but we want to see what happened on the RNAs. And actually, bacteria imply a stringent spatial and temporal control on different levels of gene expression. These mechanisms remain largely unexplored. So all of these will restrict the development of synthetic circuits. And all you need is also our feedback. First, how do we do it? The least is the advantage of our speedy back system. First, conventionally, we know that transcription and translation work continuously in the cell, but with our speedy back, we can separate these two processes by our speedy switch. And also, we can view a precise temporal dynamics in different levels of gene expression, rather than just putting emphasis on the result. And also, for doing the experiment, we can only see the average result in a cell population, but we know that there are totally different phenotypes from one cell to another. So we want to check if every single cell behaves like the average result. So with our speedy bag, we can make it. And the ends of our speedy bag system for this summer have three main points. The first is quantitative description of gene expression in both space and time. And the second, specific insight into the flow of genetic information. And the third is speedy ways to report and stop the gene expression. So if we have more precise measurements of these gene regulation, it will reveal those design rules and also lay down the foundations of complex engineered biological systems in the future. There are three devices in our speedy bag system. The first device is speedy RNA and protein reporter to locate and quantify the RNA and protein expression in a cell and also can help the promoter testing. And the second is speedy switch. If we want to view a protein expression, we can add an inducer to switch on the expression. And we create a tunable protein expression system. And also, it is a switch between the protein and RNA assay. And the last device is speedy degrader to remove the output and also can help us see the transient gene expression. So, if you fall asleep during our presentation, don't worry, just look at the right corner of our slide, you can catch our point. So let's welcome Sherry for our first device, Speedy Reporter.
Hi everyone, this is the first device in our project, Speedy Reporter. And in this device, our main purpose is to make a speedy reporter which can detect the target's location and measure its quantity. So what is the function of our speedy reporter? Our speedy reporter can express on the target immediately, and it can make the bright fluorescence comes out immediately. And this is our design. It contains a speed fluorescent protein and a speed protein with aptamer binding pair. And in our design, we fuse the two speed protein together by a peptide linker. And this is how our design works. First, we use speed protein method to make a speedy reporter. Because to generate the fluorescent protein need to wait for several hours. And the formation of the chromophore is a rate limiting step for emitting fluorescence. So in our design, we dissect the complete fluorescent protein into two fragments and make half of them contains a preformed chromophore. And when they are in spirit form, they are not fluorescent. Then we fuse them with a big aptamer binding protein, and these fusion proteins are all constitutively expressed before we use. When the aptamer is expressed, the aptamer binding protein will attach on the aptamer. And the two split fluorescent protein is close enough to each other, so they will restore their fluorescence and become a functional fluorescent protein. And in our speedy batch system, the speedy reporter can be used to two parts. First, RNA reporting part, and second part is protein reporting part. For the first part, RNA reporting part, we use EGFP as our fluorescent protein. And we choose EIF4A as our speedy aptamer binding protein. Due to it has a dumbbell structure, so it is a nice protein for domain dissection. And we select the RNA aptamer which has strong affinity with the EIF4A. And when transcription starts, the RNA aptamer comes out, and we can get green fluorescence immediately. And in our circuit design, you can use our RNA reporter to make a promoter testing. And you can insert your promoter in front of the RNA aptamer, so you can measure the activity of your promoter. And you can also insert your target gene behind the RNA aptamer, so you can make an RNA localization detecting and measure its quantity. And for the second part, protein reporting part, we use RP as our fluorescent protein. And as the same principle, you can insert your target protein in front of the peptide adapter. When translation starts, your protein will express with the peptide adapter, so we can get red fluorescence immediately. And in our speedy batch system, we also design a special device, speedy switch, to control the protein expression. And let's welcome Barry to introduce the second device, speedy switch.
Hello, everybody. Let me introduce the second device, speedy switch. One of the aims of our project is we can have the specific insight into the flow of genetic information. So, speedy switch is really important. Speedy switch provides a critical switch between RNA and protein for controlling the translation. After speedy reporter works, speedy switch will pause the translation, so we can achieve our purpose. First, we can perform the RNA assay and the protein assay in a single cell. Second, we can control the protein expression. In this device, we use the ribose switch as our speedy switch. But what is ribose switch? Ribose switch is a part of mRNA located at the 5' untranslated region. It is divided into two parts, the sensor and the actuator. And at first, the ribosome binding site is blocked. When the sensor binds to a specific inducer, the actuator will change the structure of the ribose switch, and the ribosome binding site is available, thereby regulating the gene expression. In fact, there are many kinds of ribose switches. How do we select a suitable ribose switch in our project? There is a guideline. The ribose switch and the specific inducer do not naturally exist or metabolized in the target organism. With the guideline, we can avoid the interference with the control of our ribose switch. For example, in our project, we select theophylline ribose switch as our speedy switch. We just add theophylline ribose switch will restart the translation, and the protein will be expressed. In order to do the next measurements in a shorter time, we make speedy degrader to remove the fluorescent signal. So next, let's welcome James for the third device, speedy degrader.
So let's move on our third device, speedy degrader. To make a speedy degrader, we use the accessory tag to be our tool. So what is accessory tag? Accessory tag is a short peptide which can accelerate the protein degradation. And the LVA tag is one of the most efficient accessory tag. So we use the accessory tag to remove the signals and detect the transient gene expression in our SpeedyBag system. The SSPB is a protein E. coli which can capture the protein substrate. So the CLPXP, which is a protease, can degrade the protein more efficiently. In this graph, we know that the different tag result in a different degradation time. So we can change the protein degradation time, just simply add the short specific sequence inside the coding region, and the protein will be under your control. And we can know that the LVA tag is the most efficient tag in this graph as well. So the red curve is the normal protein degradation time, and the green curve is the protein with the accessory tag. At this point, MINT stopped producing the protein. So at the left part of the line is protein production, and the right part of the line is protein degradation. We can change the protein degradation time, just simply change the tag we use. So we can make a changeable degradation time device. Let's welcome Lucy for our experiment result.
So first part of our experiment result is speedy switch. This chart shows first intensity relative time. And on the chart, we have done 6 different concentrations of self-healing and 2 negative control. And we have read from paper that if we added more than 5,000 micromolar self-healing into an E. coli, it was dead. So like this purple line, it shows like we add 10,000 micromolar self-healing into an E. coli, so there's no fluorescence intensity shown on the chart. However, the more important is that under 4,000 micromolar, our design, which uses ribose switch as a switch in translation, works. And next chart of this part shows relative fluorescence intensity versus log of self-healing concentration. And we have known that log of 4,000 micromolar self-healing is the unit of 3.6. And there is a peak at lysis, like the red circle. So the line has had a trend that the higher concentration of self-healing we added, the higher fluorescence intensity expressed by protein. And the second part of our experiment result is speedy protein degrader. So the chart shows relative fluorescence intensity versus time. And red line stands for RP and pink line stands for RP-LVA. So on the chart, we can obviously see that RP tag with LVA is degraded faster than RP, so it's work too. And this is our BioBrick part, and we do many parts in summer for iGEM. So next, welcome Clinton for our conclusion.
Hi everybody, every iGEM family. After those explanation, do you have better understanding of our project? Actually, SpeedyBag is a tool to do RNA assay and protein assay in the same single cell. If we know more inside the single cell, we can observe directly of the gene activity without the noise of the gene expression from population. In the assay, you can know the location and quantity of the RNA and protein. Also, you can have a quicker way of promoter testing. The purpose of the SpeedyBag is to explore the system much faster. Let's view the whole picture of the speedy bag step by step. As you can see, the split GFP and split EFOA is constitutively produced inside the cell. And when the main circuit transcript to RNA, they will bind to the RNA temper and causing the green fluorescence light. Through the green fluorescence, we can know the location and quantity of RNA. In addition, we can also have a quicker way of the promoter testing. By the intensity of the green fluorescence, we can know how strong the promoter is. And when the RNA migrates to a certain position, we can induce a ribose switch. And the more we induce, the more we can get the protein. This creates a tunable protein expression. In the past, it's hard to do the RNA assay and protein assay in the same single cell. But because of the conditional protein expression, doing these two assays in the same cell become reality. After we induce a switch, the translation will start. On the other hand, based on the same principle of the RNA reporter, we also want to design the protein reporter. So the split RFP and split binding protein is also constitutively exists inside the cell. When we induce the switch and the translation start, they will bind to the peptide adapter and causing the red fluorescence. Through the red fluorescence, we can track the protein you want by knowing the location and quantity of the protein. After the assay, the reporter is still there. So we want to degrade the reporter rapidly by using speedy degrader. In addition, by using the speedy degrader, we can know how the transient gene expression affect the cell, because the speedy degrader shortens the appearance of the protein appearance. And moreover, the more we induce a switch, the more protein we can get. Also, if we change the different degradation tag, we can have the different half-life of the protein. By combining the switch and the degrader, the protein expression in the cell is changeable. That is, the surface of the speedy bag contains flexibility. After viewing the whole picture, we can know that the speedy bag is a tool to look closely and quickly inside the cell in order to find more principles inside the system. And the more principles we know, the more rules we can have to design the circuit. And the more design rule we can have, the more we can do in synthetic biology. And in our speedy bag, the speedy reporter is nearly finished constructing, and our demonstration in speedy switch and speedy degrader are tested and characterized. Our future work will be combine those three devices into a system. And that's the end of the story of our speedy bag. Thanks for National Yangming University and many support. At last, if you want to explore the system, if you want to detect what's inside the black box, the only thing you need is